Hi there, this is Dr. Dan Guerra coming to you from VerEvMed and Authentic Biochemistry Studios on a semi-cloudy day on the 3rd of December. So we're going to get started right away here. Let me just minimize this and do our slideshow. This is going to be number 5 in a series of discussions on T-cell biology, particularly leading into Diaeventontological perspectives. So basically where we're going to be soon. In fact, we're going to go right into that as we speak. All right. So guys, picture outside the house here. It's the Clearwater River, just in case you want to know where we are in the Pacific Northwest. This is T-cell Diaeventontology number 5, lecture 5. The 3rd of December, there is my Facebook page where all of these are posted. There are the 2 logos for the 2 companies I'm running. That's my email address. Here are links to the podcasts and links to the YouTube lectures. Okay, so I think that's all we need to do right now. Let's get going. So I want to recap Diaeventontology because we're going to be going back to this and after we finish the complete discussion of the T-cell lineages and differentiation, various transcription factors, and cytokines, chemokine, mediated control over that regulation of T-cell remodeling and ultimately terminal differentiation in the human immune system. I'm going to take all of that information that is acting as prolegomena and put it to current published papers. A couple of them I have in mind about autoimmune responses. And of course, that's where we're going to discuss this whole concept of a 3-dimensional structural grid where genetics, the environment, and the neuroepi, that is the epigenetic immunome, are all functioning together to sculpt what possible interactions can occur with the immune system relative to normal gene expression, genes which I call now concepts because they're constantly changing, although maybe very slowly over time, ultimately leading to differential expression. Those changes in expression are, of course, the wheelhouse of epigenetics. That's why I call it neuroepigenomic immunome, right? That's the 3rd leg in that pyramidal planar discussion I have for Diaeventontology, which is going to be discussed right now, again, as a recap. So what do I mean by that? It's a term I've coined and a concept I've been generating. It's an adaptational, processive accounting of the rational human mind. So this goes way back to how I perceive how the mind is a learning event, okay? Through cellular molecular event ontology. That's my original conception, an event ontology rather than a substance ontology. If you're not familiar with that, go back and look at Whitehead's literature in philosophy. But basically, events are different than substances because we're monitoring how things change over time. And rather than looking at just matter, we're looking at how matter changes through time. And matter can include things even which are not here now. So those can be ideas or those elements of the imagination, which we use for a lot of science because we need to use ideas, and also concepts. And those concepts, as I said, could be roughly considered when you think about molecular genetics as a gene, right? A concept which itself is a movable feast. So at any rate, it's an event ontology rather than substance ontology. Okay, now substances, of course, don't just include material goods. Substances can also include ideas, right? The immunoepigenomic induction of neural, endocrine, and metallic responses, not just those, but those are 3 major ones, to the micro and macro environment. Micro or endo environment means what's happening inside the system. Through classical constitutive surveillance, this is immune response jargon here, and acquired effector cellular humoral defense stratagems using reversible, covalent, sometimes also hydrogen bonding, modification, and of course hydrophobic interactions of nucleic acids, proteins, and of course the all-important lipids. I believe this will become the central molecular theory for free will-driven agency-based existing individual adaptation and knowledge acquiring epistemology that explains the core event ontology of human existence. Now, I know that's a big for you to ask to accept the fact that I think it will become the central theory. But hopefully, as I develop it as time goes on, by using the scientific literature as my key apparatus to describe what I mean by Diaeventontology, hopefully you'll see it's pretty much commonsensical as a commonsensical way to get at published scientific research, and in a larger level to get at an understanding of the whole mind-brain problem and how existing individuals occur through time. So there has to be a mechanism, by the way, that occurs, and the mechanism has to be phenomenological, at least as we describe it in four-dimensional space-time. But it can also include noumenal aspects, so I won't leave those out, although I'm not arguing for them here. Okay. So that was the initial take on it. Here is what I call the Diaeventome or Diaeventontome. Okay. So you have the genome, you have the environment, and then you have the immunoepigenome. Okay. And you can put neuro in front of there when you're talking about any neural alterations, which of course are happening all the time, as when you're listening to this lecture. So what do I mean by all of that? Let me put on my fancy glasses. Biological networks are processes, they're not substances. They become eventual, that is, they're occurring through time, and they alter through that occurrence in nature as opposed to substantial. An event ontology that reaches across the biological networks of genetic environmental interaction is articulated and indeed synthesized through epigenetic modifications of the classical and newly emerging immune system, thus creating a responsive plastic and elastic memory field capable of metabolic scanning, focus, accession, mediation, learning, and reacting to stimuli through time. The pattern of events appear as energetic and material dynamics, much like chemical bonds. So you can look at my existing grid here, my trigonal planar grid, and these yellow bars is kind of like as chemical bonds. That is, I want to show an association, and I don't want it to be considered a loose association. These aren't just casually associated. They are bonded together and within the framework of phenomenology. So another way of looking at all this is existing individual agentically interacting, okay? So how your body involves through time and evolves over time as it relates to the vagaries of being stressed by such things as microorganism pathogenesis, such as bacterial, fungal, viral, as well as all the possible changes in cellular metabolism and mutational events, which can ultimately lead to cancer or cardiovascular disease. And then included upon that grid, maybe superimposed, is how you live, that is your behavior and your lifestyle. And that includes nutrition and exercise, right? And we all know that a major epidemic worldwide, and it is becoming worldwide because of the ready access to food with high caloric density, is the problem of obesity. And obesity is leading to a lot of these pathogenic events and frank diseases that we see proliferating and occurring at younger and younger ages, so that people live longer because we have therapies, pharmacotherapies, and neuroimmunotherapies that can alter the terminal state of having a terminal disease, meaning we don't die with it, we live with it. But at the same time, there's a lot of morbidity, right? And because epigenetics can also be inherited, yes, that's what I said, this may lead to progeny that are predisposed to epigenetic phenomena, and further leading to more pathogenesis and pathologies that we have yet to have encountered as a human system, human race. Okay, so the existing individual agentically interacts through the events obtained across the inherited genome, right? Those genes, those concepts that you've received from mom and pop, the changing environment, always changing through space and time, and a receptive architectonic, that means a globally organized, if you want a logos, neuroimmunoepigenome through time. So what are some events in physiology and pathophysiology that we can call Diaeventontological events? The molecular and cellular events that occur across mechanisms that result in real-time modifications to changing conditions. What do I mean by modifications? Modifications, for example, to gene expression, membrane turnover, membrane fluidity, things like this, things that are strictly biochemistry and molecular biology. So the mechanisms that may be involved here, but certainly aren't limited to, replication, recombination, repair of DNA, transcription, alternative splicing and translation of RNA, signal transduction pathways, including phosphorylation, ubiquitinylation, acetylation, methylation, interactions like that, which can be reversible, of course. Membrane fluid dynamics, which can also be fluid in their own right, that is changing back and forth and in other directions, non-linearly, that is stochastically over space and time at the level of microseconds. Cell fate repertoire, such as autophagy, apoptosis, various kinds of that, senescence, of the aging process, and then necrosis. And there's various types of those, like, for example, VerEvMed, which I've talked about in previous lectures online. There's also developmental programs. What's the big one we always think about? Embryogenesis, right? So that's always there, and it's being worked on by these Diaeventontological records of matter, records of substance. They're using the pejorative terms for matter and substance, meaning they count, right? Cellular division, differentiation, including all these various beautiful biochemical pathways, which we spend a lot of time on in VerEvMed, not authentic biochemistry. First of all, adipogenesis, the synthesis of adipocytes, right? Those are the lipid-containing cells in the human body. Neurogenesis, we know what that means. Myelocytogenesis, all the myeloid cell lineages. Lymphocytogenesis, all the lymphoid cell lineages downstream. Gametogenesis, of course, for reproductive concerns. Oncogenesis, and on and on and on. Okay. Oops, sorry, there's one more. So let's generate a dialectic here for T lymphocytes, okay? T lymphocytes react in the intact human dynamic event ontology, because these are part of the acquired immune system, remember, from the old terminology, still used terminology, of course, to respond to the environment, maintain cellular and physiological health, and to prepare for future change. It's a key component there too. That includes nutritional, neurological, imprinting, disease, and of course, the overarching aging. We all grow old, right? T lymphocyte lineages and associated biochemical communication are modified via changes in the epigenome, as well as in canonical, inducible, and repressible gene expression. This just has to do with repressors and inducers of genes as it relates to the promoter enhancer regions at the transcriptional level, and also at the level of chromatin remodeling, and even the epigenomic alteration of chromatin remodeling, acetylation, methylation being key features. And of course, membrane recombination. Membranes recombine is a term I've coined because when membranes are resynthesized and reorganized, they're not just made one lipid at a time, although that can occur too because of the fluid dynamic changes at the microsecond level. For example, swapping out head groups, phospholipase activities, for example, PLA1, PLA2, PLC, PLD. Those are the four major ones, right? But also just changes in acyltransferases, and then an oxygenation and oxidation of lipids within the membrane in situ, which also will alter membrane fluid dynamics in a very apparently stochastic way, because environment's impugning itself on the normal order of events, normal telos, normal logos of events. But because the environment is always there and always changing, there's a mechanism built in as well as part of the architectonics. That's, of course, what we need to study. So there are key epigenetic molecular events and dynamic processes, and those include, of course, chromatin remodeling. That's basically written large how genes are expressed, control of gene expression. CpG methylation, where the cytosine C5, the carbon 5 on a cytosine residue in a CpG island, when it's in a promoter region or enhancer region, or sometimes in the middle of exons, as long as it's an island, there's a grouping of CpGs. That methylation plays a key role in gene expression. Histone acetylation, moving from heterochromatin to euchromatin. Generally, acetylation means more gene expression globally. MicroRNA interference, right? All the different RNAs that are made, the microRNAs, the single or the small interfering RNAs, which we sometimes use in molecular biology to alter gene expression, were actually mimics of what we see, right? The interfering RNAs, right? And those small interfering RNAs are key features in how genes are expressed. And that's part of the epigenomic mechanism. And of course, the final establishment of the Diaeventontological phenomena is what the dialectic of what I'd like to bring forward here to say. The dialectic things like thesis, antithesis, synthesis, the Hegelian version of it, as read down through people like Kierkegaard and Heidegger and Sartre, but also at another level, the Platonic dialectic, which basically sets out with a form or a pre-established, understood foundational truth, and then try to tear at that foundational truth and examine it via dialectical analysis, throwing good arguments at the key features of those arguments, the premises or propositions, and then from that leading to maybe a new understanding of what those original forms were, not just in language, but also in thought. And those categories of thought are always important. Quantity, quality, modality are key features, as well as relational features. Those categories are relational features. And as I just said, quantity, quality, modality are really important to understand how science functions in the three-dimensional space and fourth-dimensional time level, and how we understand it when we're studying and analyzing the scientific literature. That's what I bring to the table. All right, so let's go back to this paper that I've been looking at through these lectures. Paper published just recently in 2019 from the Experimental Molecular Medicine Journal, volume 51. It's actually article number 80, so no page numbers are necessary. You can find it that way. So right now we're talking about NFIL, nuclear factor interleukin 3 NFIL, is also known as E4 binding protein 4. Okay, it is a repressor. What NFIL3 is it's a repressor of a bunch of different genes. Okay, right now we're locking in on the immune system. Remember, we talked about T lymphocytes. Remember, that's what we're doing this for right now. Okay, so NFIL contains a basic leucine zipper domain. So that's the kind of proteins that tend to open up and react with DNA. They have leucine zipper domains. It's a protein domain feature. It comprises certain domains in that protein, domains of amino acids between 73 and 146. And among the 462 total residues that you find in NFIL3, the N-terminal part of this domain directly binds to the DNA, while the C-terminal region of that domain is responsible for homo and heterodimerization of the protein, which facilitates its activation in the chromatin. The amino acids 299 to 363, now that's going to be distal to that domain I just talked about, comprise a transcriptional repressor domain sensu stricto. NFIL3 then represses genes by recruiting, this is what it does, once it binds, you see, the story's not over there yet, it recruits histone deacetylases and the G9A histone methyltransferase. If you deacetylate and then methylate on top of that, you shut down gene expression, basically, right? Generically, what I mean by basically. And it regulates diverse biological processes, of course, if you're changing gene expression, including circadian rhythms. So NFIL3 has been described for circadian rhythms, like in sleep. Cellular viability, the cells live or die, that autophagy, apoptosis story I was talking about a few minutes ago. And indeed, even such far-flung things as just normal metabolism in the liver, for example, hepatic metabolism. NFIL3 plays a role and has been described in the refereed scientific literature. So in immune cells, NFIL3 plays a key role in B-cell IgE class switching and the development of natural killer cells. Now, that's pretty significant, right? NFIL binds to the Ig epsilon promoter and stimulates IgE production, right? So this is often an allergic response, right? So NFIL3 is all over the map. NFIL3 deficient mice, actually, in subclinical studies, show dramatic natural killer cell loss due to the influence of that factor, NFIL3, on NK cell development, as I just was saying. Not only on development, but maturation and ultimately function, of course. NFIL3 deficient mice also exhibit the elevated interleukin 12 P440 expression in colon tissue. And when that happens, you induce Th1 differentiation, T helper cell, and that's, of course, an inflammation. Think about inflammatory bowel disease. That's right, NFIL3 is involved in inflammatory bowel disease. And that can result actually in spontaneous colitis in mouse models. So you see there. Th2 lineage cytokines are also affected by Infil3, if that's not enough, with an increased expression of interleukins 5, 13, and that's what happens in Infil3 minus Th2 cells. Okay. Furthermore, Infil3 links the circadian rhythm with immune cell development by suppressing Th17 determining factor, the retinoic acid responsive gamma T, an orphan receptor. That's a Th17 lineage often, right? That ROR gamma T is a transcription factor needing retinoic acid. And because it also uses another component in that transcription factor, not just the retinoic acid binding domain, but also what we call an orphan receptor. So it's picked up and it's used with other receptors. That's what orphan receptors means. It's a really key feature in Th17 differentiation. So Infil is hot number, right? So there it is again, nuclear factor interleukin 3 transcription factor. Aliases for it, you can see there, right? There are the various acronyms for the aliases. So it's a transcription factor recognized and binds to that sequence. Okay, that's kind of like a GATA sequence. Represses transcription from promoters by activating transcription factor sites, okay, ATFs, okay, got that? Acronyms all over the map here, you just have to follow with them. It also activates transcription from the interleukin 3 promoter, that's why that's in its name. It's a component of the circadian clock, as I said, where it actually controls the expression of the PER2 oscillation domains, if you know anything about circadian clocks. It also protects pro-B cells from programmed cell death. That is a specific form of PCD apoptosis. All right, glasses back on. Now, I want you to take a look at this overall diagram. You're looking at Infil3 expressing ID2 cells, which are also known as CHILP. Those are common helper, right? Innate lymphocyte precursors. That's what CHILPs are, okay? So you have innate lymphoid cells, right? So think about what that means. They're lymphoid, but they act like they're innate. Remember, lymphoid cells are typically T and B cells. Those are called acquired. Forget about that. Forget about that. You have gray area all over the map in the immune response. That's why now is a great time to be alive and be a scientist, because the immune system is right now being described. Every week there are new papers about it. So I want you to understand that you've got this precursor lineage here, okay? And I want you to just get this whole thing mapped out. And I'll tell you what ID, the ID is a very interesting feature, and I'll tell you what ID2 high means in a moment here. But you keep ID2 high, this is going to control gene expression in this cell lineage. You're now going to just make plain old, right, innate lymphoid cell precursors. Then you're going to start making the frank final subset differentiation. ILCs 1, 2, 3, and also this ROR gamma T LTI, which is yet another differentiation of a subset. Let's go over here and describe them. Okay. The transcriptional program that sets the identity of the ILCs, remember those are innate lymphoid cells, remains elusive. So G shows that Infil3 acts downstream of interleukin 7 signaling. This is all here. There's interleukin 7 binding to the gamma C receptor here, interleukin 7 receptor alpha. It's going to work here on the STAT5 by phosphorylating in. It's going to cause the expression of Infil3. Then Infil3 is going to cause the expression of ID2, and you end up with the precursor cells. See how important then Infil3 is to proliferate these innate lymphoid cell lineages. Okay, that's what I want you to get. And you start up with this common helper-like innate lymphoid cell progenitor. That's what those are. Okay, that's what those are. All right. So ID2 is a transcriptional regulator. Okay, it's another one. So a transcription factor, if you like. But it doesn't have that basic binding domain. It doesn't have that leucine zipper stuff, which nevertheless, it negatively regulates the basic helix loop helix transcription factors. Okay, it negatively regulates those normally proactive transcription factors, the BHLHs, right? That just describe other domain functions at the level of DNA-protein interactions, of course. And it forms a heterodimer and inhibits their DNA binding and transcriptional activity. Okay, so that's what ID2 is all about. Okay, so it's a repressor of normal gene expression at the transcriptional level by B HLH type transcription factors. There you go. It's implicated in regulating, ID2, okay, implicated in regulating a variety of cellular processes, including cellular growth, senescence, that's the aging process, differentiation, apoptosis, angiogenesis, neoplastic transformation. I'll name a few. That sounds like quite a lot, right? It actually inhibits at the specific level skeletal muscle and cardiac myocyte differentiation. That means it's related to pathologies and diseases. Okay, that's really important. It also regulates the circadian clock, as I've been saying, by repressing the transcriptional activator activity of the clock Untitled BMAL1 heterodimer. Another, if you know some circadian rhythms, you know that pathway, which is a transcriptional clock pathway for controlling diurnal fluctuations in animals. It restricts the clock and the Untitled BMAL1 location to the cytoplasm. It keeps it there. Okay, so it can't be a controlled transcription factor. Plays a role in both the input and output pathways of that circadian clock. And the input component is involved in modulating the magnitude of the photic entrainment, okay? And then the output component contributes to regulation of a variety of liver clock control genes involved in lipid metabolism. So just outrageously amazing, this ID2. You also have another component in here, this PLZF, and that gets expressed once you go from the common helper precursors to the, again, the innate lymphoid progenitors here, which is the second phase of this differentiation. So it has this PLZF transcription factor. What the heck is that? It's the promyelocytic leukemia zinc finger. So that's an old-fashioned zinc finger type of transcription factor. It's a member of transcription factor specific to the CD1D restricted natural killer T lymphocytes. So PLZF deficient natural killer T cells fail to undergo the intrathymic expansion where this is happening. Remember, T cells, thymus, right? And effector differentiation that characterize their lineage. So you don't get to make those natural killer T lymphocytes, right? Because of this PLZF, right? So if you're deficient in that, you don't get to that process. So that's showing you how these transcription factors can be swapped out in different cell lineages, something I've been talking about the previous 4 lectures. This is number 5, remember? All right. So I think we got this slide done, right? Yeah, we finished the slide. All right. Now back to just this discussion of this infill story in Tregs, okay? Let me put me over here. Here I am. Of course, infill bound to FOXP3. Remember FOXP3? That's the canonical transcription factor for what cells? That's right. I hear somebody out there in the internet saying the T regulatory cells, right? The ones that suppress inflammation. Infill bound to FOXP3 will inhibit FOXP3 target genes, because FOXP3 is a transcription factor in Tregs. What genes? Like interleukin 2 receptor alpha. Why is that important? Because you need to have interleukin 2 binding to those CD4 positive T lymphocytes so they start turning on FOXP3 expression and turn into Tregs, right? Once the FOXO13 transcription factors are regulating the early phases of chromatin. All talked about in my previous lectures, okay? So that's really important. That plays, as I say, a major role in Treg function. All this reduces, when that binds, Treg mediated immunosuppressive ability. So infill's a bad player, right? It's a bad soldier if you want Tregs to be functional. If you don't have good Treg functionality, you get runaway inflammation, including autoinflammatory diseases, where we're ultimately going to eventually, I promise. Previously in mice, it was demonstrated that the infill-3 attenuates the immunosuppressive ability of Treg cells, thus correlating with severe colitis. Okay, we said this before. Recent study shows that the methylation of the FOXP3 CNS, remember that's a non-coding region, a part of the enhancer elements expressing FOXP3. I talked about it just yesterday. And it's correlated then with Treg cell stability. This is an epigenetic phenomenon. Overexpression of infill-3 increases. Look at this. Remember, it recruited that methylation, right? All that, and deacetylation. Now we're in the epigenome domain. Okay, so it's an epigenetic phenomenon. Overexpression of infill-3 increases the methylation of 6 of those sites. That's no small change. That's going to definitely suppress the expression of FOXP3 with 6 methylations. Heck yes. One possible molecular mechanism underlying the phenomenon could be that infill-3 recruits this G9A methyltransferase, that G does stand for guanosine, and induces a repressed chromatin state at that locus. That is amazing, right? Loss of Treg stability was also observed under physiological conditions with this infill. Many studies suggested Treg instability during certain inflammatory conditions, of course, this is why it's relevant, because it's clinical now, can lead to their conversion to T effector cells, thus enhancing inflammation rather than repressing it. Okay, not good stuff. So this basic helix, loop, helix proteins represent a well-known class of transcription factors. I want to remind you of this. They regulate transcription. They act as heterodimers with a member of a class of ubiquitous proteins called the E proteins. Those are a widely expressed class of inhibitory heterodimer partners, the inhibitor of DNA binding proteins, the ID proteins. You see how this works, okay? E proteins, ID proteins, you have to keep all this, of course, well organized in your well-organized mentation. Now, more about this. Innate lymphoid cells are a family of effectors that come from the common lineage. Infil-3 is a critical regulator of them because they turn on this common helper-like innate lymphoid cell progenitor. We already talked about this. Remember this whole story. Infil-3 null mice, okay, we talked about this in detail, okay? I think this ectopic, okay, now listen to this. Infil-3 expression in lymphoid progenitors was under the control of the mesenchyme-derived hematoprotein, IL-7, interleukin 7, and Infil-3 exerted its function via direct ID2 regulation in the CHILP. So that's way back here where it's functioning. I'm getting into the granularity of what Infil is doing way back at the common helper innate lymphoid progenitor cell differentiation, beginning of that whole process that ultimately is going to lead to more ID2 formation, right? More of those kinds of cells, more proliferation of these innate lymphoid cell lineages, all of which are going to be acting unusually like innate immune responders, right? So, final thing I want to say here, ectopic ID2, when you add back by expression using a plasmid, in Infil-3 null precursors rescued the defective innate lymphoid cell lineage development in vivo. So Infil-3 as an Infil regulator is a key regulator of common helper-like ILC progenitors as they emerge during the early lymphopoiesis, which is what's going on there at the beginning of that pathway you're looking at. So just want to tell you once again, I think I've already told you what ID2 is. It's a helix loop helix. It regulates, okay, binds to the HLHs, the BHLHs. So I already told you that. All right, and I told you what E proteins are. E proteins, okay, the HLH domain of ID proteins interacts with the BHLH domain of the E proteins, right, another repressor, to form heterodimers. That heterodimerization renders E proteins unable to bind DNA. So ID2 is required for the development of natural killer cells and lymphoid tissue inducer cells. Thus, mice lacking ID2 fail to develop lymph nodes at all and include also Peyer's patches and any other secondary lymphoid organs. So this is a really key feature, right? You got to have these transcription factors. You go blocking these pathways, you lose lymphoid organs. It's got to be a major thing. All right, I just want to remind you that we got all these different T cell lineages. We've already talked about the follicular cell. We talked about Th17, 1, 2, and 3. There's Th9. We haven't talked about that yet. There's Th22. In the presence of interleukin 6 and tumor necrosis factor, you make this AHR. That's the receptor for that, making it Th22 mature cell. Key cytokine for that is interleukin 22. What is it involved in? T cell proliferation, tissue inflammation, elimination of extracellular parasites, and allergy, which is a key feature we want to look at later on. Okay, if you have the follicular cells, right, you're going to have STAT3 being expressed in the presence of interleukin 6 and interleukin 21. You're going to start making BCL6. There's going to be some interaction between the STAT3 containing cell lineage Th17s that are expressing now the ROR gamma T, right? So there's going to be interaction between those two cell lineages. But ultimately, these BCL6 cells are going to make interleukin 21. That's the B cell helper cells and humoral immunity, autoimmunity, immunodeficiency, and malignancies. It's really a key feature, right? Really important how those follicular cells are turning on B cells into plasma cells, right? And then making, for example, antibodies, right? Th17, the presence of all those cytokines, 623, TGF-beta, 1-beta, that's a really hot pro-inflammatory cytokine, the original member of that family, making the Rohr gamma T cells. Remember that interleukin 17A and interleukin 17F control the regulation of the Th17 maturation process. So that's what these dotted lines are here, coming all the way from the Tregs. This is about the tissue inflammation, bacterial, fungal, autoimmunity, and of course in cancers, really important cell lineage, something we are keeping an eye on and is being started to be looked at really importantly when looking at programmed death inhibitors, the PDL ligand system for looking at how you can jack up the T cell activation to kill tumors, which is normally trying to control that or ablate that by making those that PDL pathway tuned up and therefore cause apoptosis of the T cells, so you don't get an immune response against the cancer cells, right? So that's why they're looked at here. This is obviously coming, I should mention, from once an antigen-presenting cell is supposed to be obviously a dendritic cell because it looks like a dendritic cell, interacting with a naive CD4 positive T cell way back at the beginning. And there's all these different cytokines that they have to be involved to get to this stage. So Th1's making a lot of T-BAT in the presence of interferon gamma because it's also the one major pro-inflammatory cytokine that's made from Th1, antigen presentation, and cellular immunity. It's a really key feature for this cell lineage here, right, for Th1s. Th2, start with STAT6. We'll talk about that later on, I know, because I looked at what papers are going to contract to discuss weeks from now. In this presence of interleukin 4, you get this pathway. You get the GATA3 transcription factor, mature Th2s. That's going to make TGF-beta, which is going to control this Th9 pathway, very importantly. Also, feedback regulated by Tregs. Tregs are killing off all of these systems, see that, except for that top one. Tregs, that's what that dotted line is. Always remember that Tregs are the good guys to control inflammation. All right. And then finally, Th9 cells, STAT6, making the PU1, IRF4 transcription factors. Again, it's going to be positively regulated by TGF-beta coming from the Th2 cell lineage, all pro-inflammatory. Th2s are having humoral immunity, regulating that, of course, and again, allergy. The IL-9s are really important for the mucosal immunity, autoimmunity, again, like an irritable bowel, inflammatory bowel disease, and in cancers. And FOXP3 expression from Tregs starting off with STAT5. Again, you make mature Tregs, and those Tregs now are going to be expressing TGF-beta. And look at 35 and look at 10. These are all suppressor type cytokines, not pro-inflammatory. And it's going to be immune suppression and also basically tolerance. That's spelled wrong, sorry. All right. So, infill expression significantly increased when Treg cells were cultured under Th1 polarizing, just think about the last slide, conditions, indicating, giving the right cytokine and even chemokines, which weren't shown in that last slide, like CXCR3, that Treg cells enhance infill expression intrinsically in inflammatory conditions and decrease the stability to promote inflammation. See how that works. In addition, infill 3 overexpressing Treg cells were prone to lose FOXP3. Remember, it's going to repress that expression, and highly increases interferon gamma, and infill 3 overexpressing now Treg cells, isn't that great? Causes the suppression and causes its own expression, causes the depression of FOXP3 expression, what I meant there. Loses their suppressive function and may convert into an ex-FOXP3 Th1 cell, thereby increasing overall population of effector cells, read inflammation. It also is evident from this paper that we're looking at, this 2019 paper, that infill 3 expression is regulated by the TGF-beta signaling pathway, because that's what the data showed. I went through that data last time. Along with interleukin 2, TGF-beta is an essential cytokine for Treg cell differentiation, of course. So it's hypothesized that TGF-beta could be a negative regulator of infill 3. It was demonstrated infill 3 mediated natural killer cell development is inhibited by SMAD3. These are all transcription factors now, back to that, which is signaling molecule downstream of the TGF-beta 32 pathway. Evidence here in this paper demonstrated that TGF-beta, along with the T cell receptor stimulation, right, antigen presenting, remember that T cells only act with other cells, unlike B cells, which secrete immunoglobulins, right, antibodies. Remember that T cells, their major function in the immune system is T cells interact with other cell lineages. That's how they turn on everything. They're the fireworks factory, if you will, when you want to have inflammation. They're also part of the system that gets the fire hose out and puts down the inflammatory response. That's what T-regs job, right? Anyways, significantly reduces infil expression when you go through this pathway, while treatment with SMAD3 inhibitor restored it. So SMAD3 seems to be involved, which means TGFB is involved because SMAD3 is working downstream from TGF binding to its receptor. The reason that Th17 cells have increased levels of infil-3 even after treatment with TGF-beta is because there's another signaling pathway which props that one up. That's Interleukin 6 pathway. So Th17 cells actually require interleukin 6 cytokines for differentiation. And a previous study discovered that IL-6 STAT3 enhances infil-3 promoter activation and expression of SMAD7, different SMAD, which negatively regulates the SMAD2/3, which normally is controlling this whole process. They're putting it down. That was increased in infil-3 overexpressing Tregs, making Tregs no longer regulatory or suppressive. That all suggests that infil-3 upregulates SMAD7, and that maintains its own expression via this wonderful circular loop of controlling the SMAD2/3 by pushing that down. All right. Furthermore, the TGF-beta signaling pathway is essential for Treg cells. Infil-3 overexpression could hinder their differentiation and maintenance. Hence, this signaling mechanism explains how Tregs maintain low infil-3 expression levels. It's got to be done, otherwise Tregs no longer work. Since infil-3 is regulated by circadian rhythms, that's the members, let's superimpose a little bit of physiology here. It's possible that the frequency of Treg cells could differ according to the time of day. How about that? A recent study revealed that infil links Th17 differentiation with circadian clock through inhibition of the ROR gamma T, that, remember, is the specific transcription factor for Th17s. As this particular study here reveals that infil-3 downregulates FOXP3 expression, development of Tregs according to a time of day warrants further investigation. That's got to be a really hot topic. Remember, the paper we're talking about is 2019, summertime, so we're not yet there for how we're going to look at circadian clock regulating Tregs. But it's going to be really important, like in terms of, imagine, infection, autoimmune systems. Yeah, it's going to be key features. Maybe in pharmacotherapy, huh? Yep, especially the injectable types. You betcha. In summary, infil-3 inhibits Treg differentiation and function by blocking FOXP3 expression. Not a good thing. The study revealed that the molecular mechanisms underlying Treg cell differentiation provides information that may aid in the development of therapeutic strategies, what I just said. For immune-related diseases, autoimmune diseases included, or just pro-inflammatory responses, and of course cancer, which tends to suppress the immune response. So we are done here today. We got to where I wanted to get to. And let's get that little guy and close this big slide presentation. Bring back that. Put that there so it's not a doubling of me. So we got 42 minutes and almost 43 minutes of discussion today. I think that is just about right for what I wanted to do. This was lecture 5 on T-cell Diaeventontology. Next time, we're going to try to really move on these T-cell lineages. And ultimately, I can't promise next time, which would be lecture 6, but by lecture lucky 7, we're going to be diving into new primary literature and talk about the autoimmune system and the autoimmune diseases. So all I have yet to say is what I always say at the end of these talks. It reminds me I'm done. And this is Dr. Daniel J. Guerra saying bye for now.